The sequencing technology makes it possible to generate population genomic datasets. One important application is to compare samples with and without a phenotype of interest to find genomic features which are statistically relevant. This is called genome-wide association study, or GWAS for short. In traditional GWAS, the association power is computed for each variant individually and plotted in something called Manhattan plot. When there is a spike in Manhattan plot, scientists look for evidence of biological association. GWAS helped us to understand the causing genetic factors for many diseases, but for some phenotype, the Manhattan plot looked like this, with no clue. One possible explanation is that many genes are involved, each with a small contribution. Polygenic risk score is built based on this assumption and shown to be a more accurate genomic risk factor. Another explanation is complex epistatic interactions, where a combination of variants correlate the phenotype, but individual variants do not. The problem is they are very hard to find and only in a small subset of variants, not whole genome data. Also, there should be a large number of samples to support their statistical relevance to the phenotype. Given there is evidence for both epistatic phenotype and polygenic phenotype, why there shouldn't be an epistatic polygenic phenotype where a lot of variants contribute to the phenotype, many of those interact in a complex way? Random forest is a machine learning technique widely used in bioinformatics. It reports the importance score for variants, taking into account the interaction between them. The importance score is a perfect solution for an association study of an epistatic polygenic phenotype. But how could we apply it to whole genome biobank scale datasets? For a dataset with 100 million variants and 10,000 samples, a random forest with 1,000 trees, each with 100 nodes on average, and M-try of 1 million, we need to perform this many operations. That is a lot, even for a computer. Also, the entire 1 terabyte of dataset should stay in memory, uncompressed all the time, as the program frequently and randomly accesses it. We built VariantSpark, a scalable distributed implementation of random forest that optimally utilizes enormous available compute resources on the cloud, and it's tuned for genomic datasets. VariantSpark is faster than any other implementation of random forest and scales linearly with the increasing number of samples and variants as well as the size of the given computer cluster. VariantSpark implements several techniques to minimize slow network operations and keep all computers in the cluster working with maximum utilization. To show the effectiveness of VariantSpark, we create a synthetic phenotype for 1,000 genome dataset using PEPs. 26 variants are involved in the phenotype simulation, and there are several complex interactions between them. While VariantSpark detects 20 of those variants in a top 260 ranked variants out of 80 million variants, traditional GWAS detects only 7 of those. How to get started? Configuring a computer cluster and installing all the required software could be a complicated task. Try VariantSpark on AWS Marketplace, where with a few clicks you can get a Jupyter notebook with VariantSpark and Hail installed, backed by the number of CPU you choose. Also, there are video tutorials on our website to guide you through the steps. Thank you everyone for attending my presentation.